All right, in the last video we were talking about intermolecular forces. We got through 3 of the 4, and we ended with the idea of dispersion forces and the idea of polarizability. I actually want to talk through another example just to kind of give you some perspective, a different perspective. Last time we talked about the halogens and how the increase in size causes them to be gases versus liquids and solids. I want to talk about another example. So let's look at hydrocarbons. Anything that's just carbon and hydrogen. These tend to be anything that's just carbons and hydrogens are completely nonpolar molecules. But what we see is that if you have C5H12, that's pentane, C6H14, C7H16, and C8H18. So pentane, hexane, heptane, octane, doesn't matter. You're not taking organic right now, so it doesn't really matter what they're called. What we see is that the main difference between these is that they keep getting bigger. Carbons tend to form these long chains, these long tetrahedral chains. So this is basically a 5-carbon chain, a 6-carbon chain, a 7-carbon chain, and an 8-carbon chain. And they're all single bonds, so all the bonds rotate. So they're big, floppy molecules, and they get bigger and floppier as you go to the right. So what we expect is that because these keep getting bigger, we would expect an increased polarizability. Okay? So one way to measure this, the other consequence is that we would expect stronger dispersion forces as we go to the right because they're bigger. Okay, but one easy way we can check this, in the last example I was looking at whether they were gases, solids, and liquids. Another thing we can look at is boiling point. When something is boiling, what you're doing is you're heating it to the point where the liquid has enough energy to overcome those intermolecular forces to become a gas, so it can fly around and do its thing. So things that are lower on the polarizability, lower dispersion forces, things that are more weakly held together should boil more easily, or more likely to exist as a gas. So what we see is that this boils at 36.1 degrees Celsius. Great. We have no perspective as to whether that's a high number or a low number. That's fine. Adding one more carbon makes it more polarizable, gives it stronger dispersion forces, so the forces holding it together are stronger, so it's going to take more energy to overcome those forces, and the boiling point jumps up to 68.7. You add one more carbon, and it jumps up to 98.4. These are in Celsius, not Fahrenheit. And octane, so one more carbon, jumps up to 125.7. So you're noticing a pretty steady trend. You add about 30 degrees every time you add one more carbon to this particular type of molecule. So basically the only difference between these is more carbons makes it a bigger, floppier molecule, which increases the polarizability, which increases the dispersion forces. So nice little trend. These type of things happen all the time in chemistry, and understanding the intermolecular forces behind it helps kind of make these trends make more sense. Okay. We have talked about dipole-dipole forces, instantaneous and induced dipole forces, and then dispersion forces. Okay. The fourth one is the oddball of the bunch. It kind of is independent of the other ones. Okay. And these are hydrogen bonds. Okay. Hydrogen bonds are actually a very specific set of conditions. Okay, so what happens is you have something like a nitrogen. We have something electronegative. So things that are on the top right corner of the periodic table are the electronegative things. The closer to fluorine it is, the more electronegative it is. So this typically only really applies to nitrogen, oxygen, or fluorine. Okay, what you'll have is an actual honest-to-God bond. So like 2 atoms bonded together in a covalent bond. And this nitrogen can either be a nitrogen, oxygen, or a fluorine, but it has to be one of those 3. It's really specific. And then if you have another molecule, and you have another electronegative atom, so nitrogen, oxygen, or fluorine, let's make it an oxygen this time. What's going to happen is these things are electronegative. Hydrogen's very electropositive. So we're going to end up with a partial negative here and a partial positive here. Okay? not unexpected, stuff we talked about last semester if you had me, and if you had somebody else, hopefully you still talked about it. For oxygen, oxygen is a fairly electronegative atom, so we would expect a negative charge here. Hydrogen bonds are essentially the interaction between this positive hydrogen and the something somewhat negative on another atom. So it's this interaction right here. Okay? This is a hydrogen bond. It is a very weak force. But if you have one hydrogen bond, you probably have trillions of them. So it's one of those things that it's really weak. Each individual force is very weak, but you have so many of them that they add up to be this really dominant force. I'm going to go with the word ubiquitous. See if I can spell that. Ubiquitous means they're pretty much everywhere. Okay, I don't know why I put two parentheses, but whatever. Honestly, the analogy I use is it's like Velcro. Okay, so I'm assuming you have interacted with Velcro in your life. I'm assuming that at some point you were a little kid and you had shoes that had Velcro straps on them. I'm assuming that you have, God knows what, I don't know. Velcro is pretty common. I'm assuming you've heard of it. Velcro is really simple in terms of concept. Essentially what you have is two chunks of plastic that are really thin. One side has these little loops on them. This is the smooth side. So if you have two pieces of Velcro that stick together, one side's going to be smooth, the other one's going to be rough. The smooth side has complete loops. The other side has partial loops, which basically end up being hooks, which is why it's scratchy. And so what happens with Velcro is if you push these two parts together, this gets caught in here. And it's not perfect, and it takes no effort to rip it apart, kind of like hydrogen bonds. This is a really weak force. Each individual one can be ripped apart. And each individual hook doesn't really hold that much weight. It's not really that strong. It's not a strong force. But if you push all of them together, you can get enough hooks and enough loops that you could stick a bowling ball to the ceiling. I mean, it's adds up to be a very strong force. So here's the problem. On things like sapling homework, the American Chemical Society exam, they like to ask questions like, which is the strongest intermolecular force? I argue that it depends. Each individual hydrogen bond is by far the weakest force, but if you have a bunch of hydrogen bonds, it can add up to be the dominant force overall. So is it the weakest? Is it the strongest? It depends on who you're talking to. Okay. Hydrogen bonds usually mostly happen to polar molecules, so it's kind of like an extra force. So basically, polar molecules have dipole-dipole forces, and they're held together very strongly. But if you have a polar molecule that can also form hydrogen bonds, you're going to double up the forces. So you can have polar bonds, or you can have dipole-dipole forces and hydrogen bonds at the same time. Okay, so it's one of those ones that it's a cumulative effect. Okay, so let's kind of talk about hydrogen bonds for a minute. So let's see what we can do. Any of you who are biology majors, or people just taking biology in the background, are going to interact with hydrogen bonds. Kind of run the show as far as biology is concerned. They are stupidly important. The first example I'm going to talk about is water. Water seems like the most simple molecule. It is, the reason life exists on this planet. I'm about to take a drink because I'm thirsty while I'm talking about this, because it is critically important for my existence. It is for yours too. Please hydrate. But we almost take it for granted how water behaves. It is a super weird molecule. Okay, water forms this kind of bent structure. If you look at Lewis structures and polarity, this is the stereotypical polar molecule. So it is partially negative where the oxygen is, and the average between the two hydrogens is somewhat positive. Okay? And so, if you had a bunch of water molecules, which is not hard to come by, sorry for using plastic, the water here tastes bad. What you'll find is that there are dipole-dipole forces. And that's when it's in the liquid form, the dipole-dipole forces are the dominant force. So basically this is polar, but it can also form hydrogen bonds. Okay. So the polar force, when it's a liquid, is the dominant force. Okay. When it is a steam, honestly, it's a gas and they're not really interacting. It kind of overcomes all these forces and it splits apart. Okay. Hydrogen bonds actually tend to be the dominant force when it's frozen, when it's a solid, when it's ice. Okay. And here's why. What happens is these things, this is my water molecules of oxygen, 2 hydrogens. They, as a liquid, they flop around, they do whatever they're going to do. But basically, if push comes to shove, negative, positive, negative, positive, they line up like this for the most part. Okay. If it freezes, water is one of the only things in the universe that expands when it freezes. Most things contract. The reason it expands is because it's trying to line up hydrogen bonds. So what we have is an oxygen, which is electronegative, and then we have a hydrogen, so there's an actual bond. If we had another molecule of water, it's going to force itself to line up like this. And we end up with that hydrogen bond. And the beauty is that water can do this twice. So each one of these can do this over and over and over again. And then you'd have another hydrogen up here, and it just goes on forever. And this is how you end up with sheets of ice. This is how the crystal structure forms. But it has to go from being on top of each other like this, it has to literally pull apart. Can't make my hands fit in here. It has to pull apart to get that hydrogen bond interaction. Okay, which is why ice, like if you take water and you put it in your freezer, it's actually going to expand to freeze. It has to pull apart to form these hydrogen bonds. This is also why ice is slippery. Most things when they freeze are not slippery. Ice is the oddball of the bunch. It's January outside. Well, honestly, if you're watching this the first day of class, it's probably February. Either way, there's probably ice outside. You know that when you step on it, it's slippery. The reason it is slippery is that it's self-lubricating. What that means is this is ice, and when you step on it, these bonds are easy to break, okay? Because hydrogen bonds are a very weak force individually. So what happens is when you step on this, it squishes these together and it actually forces it back into the liquid form. So you have this sheet of ice, and up on the top, right where you push down, water forms just long enough to make it slippery, and as soon as you release the pressure, it goes back to forming these ice crystals again. These things are easily formed, easily broken, okay? This is unique stuff. This is, I don't want to like dwell on this too much, but water is a super weird molecule, and it's mostly because of the presence of hydrogen bonds, okay? Another example is DNA. I am not going to draw the all the different base pairs of DNA. But essentially, if you look at DNA, I'm going to draw a really bad DNA. Okay, yay, crappy DNA. You have 2 strands, double helix. It's my attempt at a helix is just a swirl. What you find is that these base pairs are specific. You know how they say that there's like the base pairs that line up together, like the A and T and G and C and all that stuff? The reason they line up is that it allows nitrogens and oxygens and hydrogens to form hydrogen bonds. So literally the only thing holding DNA together, holding these double strands together, are hydrogen bonds. And the beauty of this is because your DNA is really long, there's so many of these that DNA is a very stable structure, okay? If you want to replicate DNA, what happens is a ribosome starts at one end and it basically pulls this apart, which is easy to do because each of these individually comes apart. And what you'll find is that it'll replicate one side, and then as it lets go, as it passes through, they'll just snap back together. It's like Velcro. It's like if you have a long strand of a thin piece of Velcro, you can open up one end, put your finger between the two, and then pinch it back together, and you can slide your finger all the way through. And each of the individual hooks and loops come apart really easily, but if you just push them together, they come back together. That's how DNA works too. Ribosomes will break each of these individual bonds without hardly any effort at all, replicate what it needs to, and then as it passes through, these things happily snap back together. So it comes out the other side, it's able to technically rip this in half without completely ripping it in half, because this thing comes apart really easily and goes back together really easily. Hydrogen bonds are the reason you're alive. Okay, I don't want to oversell this, but hydrogen bonds are like critically important for biology. When you have DNA, or when you have protein structures, you have your amino acid strands, and then they fold up to form this tertiary structure. The tertiary structure a lot of times is held together by hydrogen bonds. So you have this huge huge protein sequence, and most of those amino acids in there really aren't that important, except that when it folds up to form this like glob shape, there's enough interactions in there that it folds onto itself, and the folding of a protein is based off where the hydrogen bonds are within one molecule. So it's not even necessarily between two molecules like this, it's actually one molecule hydrogen bonding to itself. Okay, so if you're taking biology, if you're a biologist at all, this stuff comes up all the time. It's critically important to understanding how the world works. All right, thanks.